Hello, everyone. This week we're going to talk about oxygen requirements. And again, we're still talking about microbial growth conditions, and specifically, we're going to talk about those oxygen requirements of bacteria. So we can categorize bacteria actually based on their oxygen requirements. The first group of bacteria that we're going to talk about are obligate aerobes. Obligate meaning a must. So obligate aerobes, for them to grow, they must have oxygen. So they require oxygen. They cannot survive without oxygen. They must have oxygen. There's another group called obligate anaerobe, and they are the exact opposite. They cannot tolerate oxygen. Oxygen for them is poisonous. So they are going to steer away from oxygen. They are only going to grow in the absence of oxygen. We also have a third group called aerotolerant anaerobes, or sometimes they just call them aerotolerants. And those are the ones that can grow in aerobic conditions, meaning like in conditions that have oxygen, or in anaerobic conditions, conditions that don't have oxygen. The thing is, they don't choose oxygen. So whether you give them oxygen or you eliminate it out of the environment that they are living in, it's not going to make a difference for them because they don't even use oxygen in the first place. And so for them, again, oxygen will not be used. So, whether it's there or whether it's not, I mean, they're not harmed by oxygen or anything. And so at the same time, as I said, they don't even use it. So they're going to grow, as I said, either in aerobic or anaerobic, and they're not going to have a preference. Okay, so they don't have a preference to aerobic conditions versus anaerobic conditions. They have absolutely no preference. The ones that do have a preference are called facultative anaerobes, and they prefer oxygen. So they prefer oxygen, but if they are placed in conditions where there is no oxygen, they're still going to be able to grow. But they do prefer oxygen. And that's the biggest difference between aerotolerance and facultative anaerobes, is that when you take a look at the tube and you take a look at the turbidity to kind of figure out whether the bacteria is aerotolerant or facultative anaerobes. Aerotolerants, they're just going to grow throughout the whole entire tube. Facultative anaerobes, even though they're going to grow throughout the whole entire tube, you're going to notice more growth towards the top of the tube where there is more oxygen. Now, we did talk about how we use deep, right, the deep media to figure out based on the growth pattern, whether it's obligate aerobe, anaerobe, etc. Now, other than using the deep, there's another medium that we use, but this one is actually a broth. So it's not a deep, it's actually broth, so it has 0 agar, but it's a special type of broth called thioglycolate broth. And it's a reducing medium. It has a chemical known as sodium thioglycolate. And so that's why, again, as I said, we usually call it the thioglycolate broths. And this chemical basically reacts with oxygen and it eliminates it. Okay. Why do we use thioglycolate broths instead of deeps? The main reason why we use these type of broths is to help us culture anaerobes, to help us culture specifically obligate anaerobes. Remember how I said obligate anaerobes? They require 0 oxygen. They don't tolerate any amounts of oxygen. So it's kind of hard to grow, okay, and culture basically these obligate anaerobes. Why? If you think about it, everywhere there's oxygen, right? So for those bacteria that do need oxygen, So obligate aerobes, facultative anaerobes, they're fine because there's oxygen everywhere. Even in the incubator, by the way, there is oxygen. So they're just going to do absolutely fine. The ones that we're going to struggle culturing are the obligate anaerobes because, as I said, oxygen for them is poisonous. Well, there is a problem because oxygen is found everywhere, including the incubator. And so that's why we go ahead and we use the thioglycolate broth because it's going to help eliminate the oxygen. And it has a screw cap that you can go ahead and screw on tightly, to prevent any oxygen from entering into that tube. But the thing is though, is this tube going to have zero oxygen? No, there is still going to be some oxygen, okay? And actually there's an indicator that would kind of tell us how much oxygen is in there. Do you guys see this like pink color right there? So there's also an indicator in this broth, and this indicator actually turns pink when there is oxygen. So of course, the less pink you see, that means the less oxygen there is. And do you see how the pink is mainly up here? What does that tell you? It tells you, okay, there is still some oxygen, but it's mostly up there. So this whole entire part that's pink, that would tell you that there is oxygen, okay? And so notice how down here there is no pink color. So it would tell you that this is the region where there is no oxygen. So where do we expect obligate anaerobes to grow? Well, we expect them to grow basically in this whole entire region right here. See this whole entire yellow part? And that's where there is absolutely no oxygen. So you can actually have obligate anaerobe growing throughout this whole entire area. Okay, up here, this is the part where there is oxygen. Now, if it's an obligate aerobe, it's going to be at the very, very top because obligate aerobes, they definitely want oxygen. So they want, like, to kind of go up there and get as much as they want. Facultative anaerobe, you're going to find it growing or, cultured throughout the tube, but they're definitely going to be more growth up at the top because they prefer oxygen. Aerotolerant, the whole entire tube is going to be turbid, and you're not really going to see a difference whether it's turbid up here or down here because, as I said, aerotolerants, they don't even use oxygen. So, but anyways, the main reason, again, you guys, that we use thioglycolate broth is to help us culture obligate anaerobes, okay? Now, how do we usually inoculate a bacteria in a thioglycolate broth? We use an inoculating loop. Of course, after you sterilize your loop and then you use it to pick a sample, of a bacteria colony, you go ahead and you kind of stick the loop, you stick it all the way in, but don't vigorously shake your tube, okay? So don't shake your tube, don't even mix it, okay? So don't kind of like, flip your tube, upside down to kind of mix the bacteria that you just deposited in there. Don't do that because if you shake it or mix it, you're going to introduce more air, and air has oxygen. So you're going to introduce more oxygen in this broth. And remember how the whole reason, again, why we're mainly using this type of broth is to see if we have obligate anaerobes, or to help us kind of like, culture obligate anaerobes. So that's why you don't want to shake, you don't want to mix this tube at all. So we usually stick in the loop in there very gently, and you just kind of pull it out very gently. So how am I going to mix the bacteria that I just deposited? We usually take the tube and very gently kind of roll it between the palms of our hands for a few seconds, or for a few times, and then you go ahead and you incubate your tube. And then again, once you pull it out of the incubator, again, make sure that you don't shake or mix it, okay, when you get it out of the incubator. And then you take a look at the location of the bacteria growth, and based on the location, if it's mostly up at the top, obligate aerobe. If it's mostly towards the bottom, obligate anaerobe. If it's throughout, but there is more growth up at the top, that would be facultative anaerobe. If it's throughout, without preference, okay, in the part that has oxygen or in the part that doesn't, that would be aerotolerant. And then again, here are some cartoon images to kind of show you that up at the top would be obligate aerobe, towards the bottom, obligate anaerobe. And again, obligate anaerobe, oxygen for them is poisonous. That's why they are steering away from the oxygen. Facultative, as I said, you would find it throughout, but notice how they have a preference, okay? So therefore they prefer the region with more oxygen. Aerotolerance, they don't have preference for oxygen because they don't even use it, okay? So they can just grow throughout. For this lab, you're going to go ahead and, do that a virtual lab. And so you have a bunch of different types of bacteria over here. And so you're going to inoculate. Here's the inoculating loop. Of course, you sterilize the inoculating loop, dip it into the bacteria, and then you're going to go ahead and dip it into the thioglycolate broth. Again, that pink color is not indicating bacteria growth. That pink color is just indicating that there is still some oxygen. Remember, that broth has an indicator, and it turns pink in the region that has oxygen. So notice how, again, the pink is only up at the top, meaning that this whole entire area, okay, that's yellow, it's actually free of oxygen. And so therefore, if there is actually an obligate anaerobe, it can grow all the way actually up to here, since this whole thing, doesn't have any oxygen, okay? And again, how do I know that this whole entire part has 0 oxygen? Because I don't see that pink color, okay? So see how the thioglycolate broth can actually help us eliminate oxygen, and it will, kind of definitely also help us out determine the oxygen requirements for our bacteria. Now, to culture obligate anaerobes, other than using the thioglycolate broth, we can also use what's known as the Brewers Jar. And this is how a Brewers Jar looks like. You go ahead and you place your plates in this jar right here, and then you add a packet, and this packet has a bunch of chemicals in it that eliminates the oxygen, and then you go ahead and you actually also seal it. So you ensure that there's absolutely no oxygen whatsoever in this jar. And so the environment in the brewer's jar would be an anaerobic environment, and so definitely obligate anaerobes can grow in there, can get cultured. Now, what else can actually get cultured in an anaerobic environment in a brewer's jar? Well, facultative anaerobes. Even though facultative anaerobes, they prefer oxygen, but they are still going to be able to grow in environments that have no oxygen. Aerotolerance can also grow in an anaerobic environment. The only microorganism or the only bacteria that's not going to grow in a brewer's jar, out of the ones that we've mentioned so far, would be the obligate aerobe. Okay, because obligate aerobe requires oxygen, so it will not, be cultured in a brewer's jar. Now, there is another environment that we can create, and that would be a capnophilic environment where the amount of oxygen is low and the amount of carbon dioxide is high. And we call this setup the candle jar. So what we usually do is we get a jar, you're gonna stack your plates in here, and then to the sides, to the plates or on top of your plates, you go ahead and you actually put a lit candle in there. Now, for that candle to stay lit, for that flame to stay going, you need oxygen. And so what happens is once you go ahead and you place the lid, and you go ahead and you close it up, well, this flame is going to burn out. And so when it burns out, it's basically going to release carbon dioxide. So in this environment right here, the amount of carbon dioxide is going to be pretty high, and the amount of oxygen is going to be low. So in a capnophilic environment, since there is still oxygen, obligate aerobes can still grow. Okay, so even though it's not a lot of oxygen, obligate aerobe just requires oxygen, so it's still going to be able to get cultured in there. Facultative anaerobes will also be cultured. Aerotolerance, because aerotolerants don't choose oxygen, so they're going to be able to get cultured. What's not going to be able to get cultured in a candle jar, again, out of the ones that we've listed, would be the obligate anaerobes. Because obligate anaerobes requires 0 oxygen, and there is a bit of oxygen in the candle jar. The incubator has the same levels of oxygen as the atmosphere. So incubator, definitely obligate aerobes will be able to get cultured, facultative anaerobes as well. Aerotolerance, because they don't even use oxygen, and whether there's oxygen or not, they're just going to get cultured. And so what's not going to be able to get cultured, in an incubator, if you don't use the bioglycolate broth, it would be the obligate anaerobes. So, by the way, obligate anaerobes, when you go ahead and you inoculate it in a thioglycolate broth, we do stick it in the incubator. But the reason why it works, right, even though the incubator has oxygen, it's because the thioglycolate broth has, the chemical that would eliminate the oxygen, okay? And so that's why if we stick it in the incubator, it will still, work. But let's say you stick a regular, nutrient broth, not a thioglycolate broth, in the incubator, the obligate anaerobe will not be cultured, okay, because there's tons of oxygen. But if you have a tube, a thioglycolate broth, you inoculate it with bacteria, stick it in the incubator, then yes, the obligate anaerobe will be able to get cultured. But if you're not told specifically that a thioglycolate is being used and being incubated, then assume that the incubator is not going to allow the obligate anaerobes to be cultured in it. Okay, so you must be told that the thioglycolate broth is put in the incubator for you to say that an obligate anaerobe might be cultured in there. And then these are some results that you're going to go ahead and analyze. So test tube A, and then like here's B, C, and D, and then you're basically kind of trying to see the turbidity, right? So based on where the turbidity, okay, is located, and that's why, as I was saying, we never shake, right? Never shake or mix those tubes, because you don't want to mess up, basically this pattern that you have right here, okay? And then those are also another set of, plates. And this plate right here was placed in an incubator. This was placed in a candle jar. So just to kind of refresh your memory, incubator, there's lots of oxygen in an incubator. Candle jar, there's a low amount of oxygen, but high amounts of carbon dioxide. And then brewer's jar, this plate was placed in a brewer's jar where there is 0 oxygen. So based on those 3 conditions, 0 oxygen, low oxygen, high oxygen, you're going to take a look at the growth, okay, for each bacteria. So, and then determine whether this bacteria is facultative anaerobe, whether it's obligate, obligate aerobe, obligate anaerobe, etc. So, for example, you're going to take a look at M. luteus, and you're going to see, okay, it's cultured when I placed it in incubator. It was cultured when I placed it in the candle jar. but when it was placed in the brewer's jar where there's 0 oxygen, it was not able to grow. And so based on this information, what do you think M. luteus is? Is it obligate aerobe, obligate anaerobe, facultative anaerobe, or is it aerotolerant? And you're going to do that with each single, bacteria. Compare, again, all 3 plates, and based on the results, figure out their oxygen requirement.